And as I was saying, we'll finish up by talking about the dangers of inbreeding. And to handle this, I need to introduce just a couple more mathy concepts. And this is mostly the last that we'll do for a while, certainly before the first exam, which is tomorrow, don't forget. Um, we can define alleles in two different individuals in a population as identical by descent if we both got them from a recent common ancestor. Um, Quick example. As I mentioned before, eye color in humans is actually governed by several genes, but to a rough approximation, brown is dominant to light. I have brown eyes. My sister has brown eyes. My mother has light eyes. So the only way we could have gotten our eye gene or eye color is from my dad, who's got brown eyes. So my eyes and or my brown eye alleles and my sister's brown eye alleles are identical by descent because we both got them from my dad. In fact, we know that my dad's mother had light eyes, so my dad must be heterozygous for eye color, one brown and one light allele. And since both I and my sister have brown eyes and my mom has light eyes, the only way we could both have brown eyes is if we both inherited the same dominant brown-eyed allele from my dad. On the other hand, as far as I know, I've never gotten up real close, but as far as I can tell, the Emperor of Japan has brown eyes. However, our alleles are not identical by descent because we don't have any recent common ancestry. Now, we have distant common ancestry. If you go far enough back, somebody worked out that the last human being who was a common ancestor of everyone alive lived something on the order of 6,000-ish years ago, roughly. Human populations have gotten around an awful lot more than we sometimes give them credit for doing. So if you go far enough back, the Emperor of Japan and I have a very, very, very distant common ancestor, but certainly no recent common ancestry. You know, I have no known relationship to anyone who's Japanese, much less the royal family. So that's what we mean by identical by descent. The fraction of alleles in a population that are identical by descent is called the inbreeding coefficient. And we represent that as capital F. And we derive it like this. Go back to that oyster example that I always do and imagine that you have this pool of eggs and sperm being randomly drawn. The probability of getting a homozygous zygote with the alleles identical by descent is PF. P is the probability that you're pulling out a gamete and you're getting big A. And capital F is the probability that you're pulling out a second gamete and it will be identical by descent with the allele that's in the egg. And the probability of getting a big A zygote with alleles that are not identical by descent is P, which is the probability that the egg will have the big A allele, times P, which is the probability that the sperm will have a big A allele, times 1 minus F, which is the probability that the egg and the sperm alleles won't be identical with each other by descent. And we can use the same reasoning to show that the probability of getting a little a zygote with alleles that are not identical by descent is Q squared times 1 minus F. And the probability of getting a heterozygote is 2 times P times Q times 1 minus F. So the total frequency of big A genotypes in a population is the sum of the two previous formulas. It's PF plus P squared 1 minus f, and we can rearrange that a bit, and we can turn that into p squared plus pf 1 minus p, and 1 minus p happens to be q, so we can rearrange that as p squared plus pqf. Notice that if there's no inbreeding in the population, then the frequency of big A genotypes is just p squared. That's all. If there is inbreeding in the population, then the frequency of, there. If there's no inbreeding, then that's the frequency of big A homozygotes. That PQF is the frequency of extra homozygotes. If there's no inbreeding, then big F is 0 and this disappears. Right? If there's no inbreeding, then F is 0 and we can X out that little term. If there is inbreeding, then what this gives you is more homozygotes than you would expect if the population is in Hardy-Weinberg. In fact, that's the immediate sign of a population that's inbred, is you have more homozygotes and fewer heterozygotes. Intuitively, this should be pretty clear. If an individual mates with a close relative, right? The individual that does that, has a certain number of alleles. An individual that's closely related is more likely to be carrying the same alleles. So if I will momentarily suppress my nausea, let's say I have some recessive allele and my sister, and I have kids with my sister, which I wouldn't do because I'm not from Marshall, Arkansas. But if I have some recessive allele and I have kids by my sister, the odds are greater that my sister is also carrying that allele, for the simple reason that we're related, which means we share a lot of our alleles. So if I have a recessive and I have kids with my sister, the kids are likelier to be homozygous for that recessive allele than they would be if I had kids with someone who is not kin to me at all. Okay, so if you reproduce with a close relative, you have greater chances of producing homozygotes. Increased homozygosity is the signal for a population that's undergoing some inbreeding. We can use the same math to show that the frequency of little a genotypes is going to be q squared, which is what it would be under Hardy-Weinberg, plus PQF, which is the extra homozygotes that you get if there is significant inbreeding. And the frequency of big A genotypes works out to be 2PQ 1 minus F. Okay, so how do we calculate F? That's often what we really want to know. The observed frequency of heterozygotes we can call H sub O, H observed, and that's 2PQ 1 minus F. If we solve for F, we get F is equal to 1 minus HO divided by 2PQ. 2PQ is the expected frequency of heterozygotes if the population has no inbreeding. And we can call that H sub expected. So we calculate F as 1 minus observed heterozygotes over expected heterozygotes. Think about this. If the population is in Hardy-Weinberg equilibrium, the observed number of homozygotes should be equal to 2PQ, which is the expected number if the population's in Hardy-Weinberg. If the observed is equal to the expected, then this fraction becomes a number divided by itself, which is 1, and 1 minus 1 is equal to 0. So if there's no inbreeding at all, HO equals 2PQ, this fraction becomes 1. 1 minus 1 equals 0. F is going to be 0. If there's lots of inbreeding going on, then the observed right here is going to be very small. It's going to be smaller than 2PQ. So if HO is very small, HO divided by 2PQ is very small, and F will be 1 minus something small, meaning F is going to be close to 1. That's the equation that I want you to know. F is equal to 1 minus observed heterozygotes divided by expected heterozygotes. Let's go ahead and I'll work one problem with that just to try to drive that home. So here's our practice problems. And the one I wanted to work is, oh, that Mama Chog one's a beast. I want to do, no, let's do Avena fatua. Okay, that's wild oats. And Jane and Marshall studied wild oat population genetics in California, and they looked at the hairiness of the leaf sheath. If you're a botanist, hairiness is called pubescence. Yes, that is related to words like puberty. That's the age at which you get hairy, as it were. And it happens that that's controlled by a single locus, 2 alleles, and here's the frequencies of genotypes in one population. 57.1% big L, 7.1% big L, little l, and 35.8% little l. So we want to calculate the allele frequencies, predict the frequencies if the population's in Hardy-Weinberg, and then calculate F. Okay, how do we do that? Let's go over to the whiteboard. There is our whiteboard, and let's clear that. And here is our data. So the first thing we want to do is calculate the allele frequencies. Okay. Well, I'm going to turn all of these to decibels. 5, 8. Okay. How do we calculate allele frequencies? Well, frequency of big L, if you want to do it the easy way, it's just equal to 0.571 plus 0.071 divided by 2, which should be equal to, one momento, 0.571 plus 0.071 divided by 2 equals, that's not right, 0.175 by that is 0.61. That's the quick way to do it. If you want the long way to do it, vanish those decimal points and pretend we have a population of 1,000 individuals. 571 are big L, and 71 are big L, little l. And you get 571 times 2 plus 71 times 1 divided by 2,000, the total number of alleles in the population, and you'll get the same answer. And the frequency of little l is going to be equal to 0.358 plus 0.071 divided by 2. And that should be equal to 0.39. Okay, there we go. We've got our allele frequencies. If we're in Hardy-Weinberg, the expected frequencies are going to be the expected frequency of the big L genotype is going to be 0.61 squared, which is going to be about 0.37. 0.61 times 0.61 equals, yeah, is going to be 0.372. And the expected heterozygote frequency is going to be 2 times 0.61 times 0.39. 39 equals 2 times 0.61 times 0.39, which is equal to 0.476. And little l, our expected frequency is 0.39 times 0.39, which is equal to, it's going to be about 0.15. It's going to be 0.152. 0.152. Add those up and we get 0, 9 carry, 7a, 0.99. Yeah, we get close to 1. So we did the math pretty much right. So our observeds are up here, our expecteds are up here, and you may well have noticed that that's our observed heterozygote frequency. That's what we'd expect if the population is in Hardy-Weinberg. We are way the heck out of Hardy-Weinberg equilibrium, right? Those observeds do not line up at all with the expecteds. How do we calculate the inbreeding coefficient? Well, F is just equal to 1 minus H observed divided by H expected. And so that's going to be equal to 1 minus, our observed is 0.071. Our expected is 0.476. And that sum, whoops. Okay, hang on a minute. Okay. That sum is going to be .85. And that's our inbreeding coefficient, 0.85. 85% of the alleles that are floating around in this population are identical with other alleles by recent common ancestry. That's a population that is very heavily inbred. My guess is that what's going on here is this must be a plant that commits a lot of the ultimate in inbreeding, which is breeding with itself. That's certainly something I would suspect. A lot of plants can self-fertilize, whereas it's much less common in the animal kingdom. But that's how you do that. You measure the observed heterozygosity. You estimate heterozygosity under Hardy-Weinberg. If these two numbers were the same, then HO would be equal to HE, and this fraction would be equal to 1, and F would be equal to 0, or very close to 0. If HO is very close to HE, then this fraction is nearly 1, and 1 minus something that's almost 1 is almost 0. So under Hardy-Weinberg conditions, F should be 0. As it is, F is quite high because we have a very a big deficit of heterozygotes observed compared to what we would expect. So that's how you handle inbreeding problems. And I think I have equipped you with everything you need to know to solve the rest of the practice problems. Yay, right. Okay, let me do new share and there, sharing that. Hopefully everybody can see this now. So the formula that I'd want you to remember, the formulas that I want you to remember are, the Hardy-Weinberg formula, and then fitness added to that, and this, and that's it. There's only a couple of mathematical equations I would expect you to have at your fingertips. Here's the thing about inbreeding. Let's get away from numbers and look at cute furry animals for a while. Inbreeding increases homozygosity in a population. And again, that's because if you mate close relatives, they are very likely to be carrying the same alleles because they're close relatives. And here's the thing, if you're an animal breeder or a plant breeder, variability is your enemy. You don't, if you're breeding horses and you want to breed ponies, and everyone knows you as a breeder of good ponies, and you have customers lined up to buy your ponies, you don't want to crossbreed two ponies and get a Clydesdale. What are you going to do with that? If you're breeding fast racehorses, a lot of money is riding on those because, gambling makes that big business. So you've got two really fast racehorses. They win the Kentucky Derby and the Preakness and all of that. All of your horse breeding friends are practically throwing money at you for the right to buy their offspring. You don't want to mate two fast racehorses and get a Shetland pony or a Clydesdale. If you're breeding fast horses, what you want is lots and lots of fast horses. Same for plants. If you're breeding big tomatoes, you want lots and lots and lots of plants for many, many generations that will all produce big tomatoes, right? What you want is predictability. And the way that breeders have worked ever since well before Darwin's time was to use inbreeding to fix desired traits. If you've got a very fast horse and you want to make damn sure that all of the offspring are fast horses, mate that fast horse to his own sister. Why? Because if that fast horse has alleles that make it fast, the horse's sister is likeliest to also have the same alleles. So you should get offspring that are homozygous for all of the fast alleles. And that gives you offspring that are themselves very fast, which is what you want because you want to be a successful racehorse breeder. And this is what's at the root of an awful lot of our domesticated varieties of animals and plants. You pick out individuals with traits that you like. And if you want to get lots of individuals with the same traits that you like, you breed the ones that you've got with their siblings, and you get populations with all the traits that you want. The downside of this is you also end up with populations that have all of the traits that you don't want. An inbred population can be homozygous for the genes that you want, but it will also become increasingly homozygous for genes that you don't want. The most obvious case of this is genetic diseases that you get in breeds of dog. A purebred Doberman Pinscher just means an inbred Doberman Pinscher because all Dobermans descend from a small number of founders. Thoroughbred racehorses, it's even more extreme. If by definition, a thoroughbred racehorse is descended from one of three Arabian stallions that were sent to Britain, that were called the Godolphin Arabian, the Byerly Turk, and a third one whose name I can't remember right now. But there were three stallions who were bred with something like 8 mares. and their offspring were bred together, and those offspring were bred together. All thoroughbred racehorses today come from those 3 stallions and 8 mares. In fact, that is the legal definition. You can't be a thoroughbred horse if you don't have a pedigree that takes you back to that founding pool of 3 males and 8 females. And that means that thoroughbred racehorses are very heavily inbred. They're homozygous for all of the genes that you really want in a fast racehorse, but they're also homozygous for things that you don't want. I'm not familiar enough with thoroughbreds to name those, but if you look at dog breeds, it's the same thing. Dog breeds come from small founding populations that have been heavily interbred with each other. Doberman pinschers have a high frequency of a type of hemophilia called von Willebrand's disease. One of my professors in grad school kept Dobermans, just loved them, said they were terrific dogs, but the dogs he adopted were VWD positive. They had this type of hemophilia and required special care. Dalmatians, very high incidence of congenital deafness in Dalmatians. Boxers are very prone to cancer. There's others that I can't call to mind, but anybody that's got experience with purebred breeds of dog may be familiar with things like this. And the way animal breeders deal with this is by inbreeding coupled with very strong selection. If you're a Dalmatian breeder, you are expected, I think it's part of the code, other breeders won't deal with you if you don't do this. Every dog that shows signs of deafness is supposed to be put down. You can't even, or you're not supposed to anyway, spay or neuter a deaf dog and give it away as a pet. There are families that might be perfectly happy with a deaf Dalmatian and, not mind caring for it at all. But the code of ethics among Dalmatian breeders is that every deaf dog must be euthanized. And so what that will eventually produce is a breed of dog that's homozygous for everything you want because of inbreeding and doesn't have any traits that you don't want because of selection against them. It's not necessarily really healthy for the dogs, but it's the way that animal breeding works. And here's a recent example. There's a regular tiger on the left and a leucistic tiger on the right. These white tigers are not technically albinos. They produce dark pigment. They just don't produce orange. And this is called leucism. And wild white tigers do very rarely show up as mutations. The last wild caught white tiger was captured in India in 1951, and he was named Mohan. I'm not sure what that means. And I found a bit of his pedigree chart online. Here's Mohan. He was mated with an orange female. There weren't any white females known. He was mated with a healthy orange female named Begum. And here's all their cubs. I'm sure you've seen pedigree charts before. So this cub right here is presumably heterozygous, but was orange. These are some more offspring of theirs over here. These are some more offspring of theirs down here. And then Mohan comes all the way down here and is mated with his daughter, Radha. And Mohan's white, Radha is heterozygous. So all of the offspring came out as white, came out as leucistic. Here they are, Sukeshi, Mohini, Rani, and Raja. Sukeshi got mated to granddad, Mohan again, and they had lots of kids, and those kids are down here. Here's Gomiti and Gautam and Govind and Lakshmi and some more and so on. So the last wild caught white tiger was bred to a female and then bred to his daughter and then bred to his granddaughter. And what that gave you very quickly is a population of homozygous white tigers that could mate with each other and produce more white tigers, because that's what you want if you're in the tiger business. You want predictability. And that lineage has been maintained ever since by zoos and circuses and animal breeders and um, uh, um, shoot, what's her name? Sorry, I was going to make a joke about that reality show about tiger breeders, and now I can't even remember what. Oh, I'm sure Carol Baskin. Actually, Carol Baskin probably hasn't, and here's why. The problem is that Mohan's descendants are homozygous for white fur, but they're also homozygous for other things. Here's one of Mohan's proud descendants with rather severe facial deformities, for example. So this one is not just homozygous for white fur. He's evidently homozygous for some kind of allele that messes with facial development. Um, and if memory serves, I don't think this tiger's all that mentally sharp either. And this is why the Association of Zoos and Aquaria, the AZA, banned its members from breeding white tigers in 2011. White tigers may be homozygous for white fur, but they're also increasingly homozygous for alleles that give them a lower quality of life, cause them to suffer. You'll still see white tigers because, you know, there are circuses and roadside zoos and, you know, private tiger breeders. You know, who was Carol Baskin's enemy? He'd probably breed white tigers. Anyway, there's members that don't follow that code, and they're not part of the Association of Zoos and Aquaria. So white tigers have not died out, but reputable zoos and aquaria no longer breed them. They may have some on display, but they don't breed them to each other. This breed of tiger needs to be allowed to die out. But sometimes even that tendency to disease is exactly what you want. If you go on to research in cell biology or physiology or neuro, Dr. Padberg doesn't work on mice as such, but Dr. Clancy, who was retired a few years ago, certainly did. And she did a lot of research on inbred mice. There's a lot of medical research that's done on inbred mice. And inbred mice are strains of mice that have been mated brother to sister for at least 20 generations and sometimes many hundreds of generations. Sometimes it's a lot more than 20. That level of inbreeding over that length of time makes these homozygous at practically all loci. Members of an inbred mouse strain aren't clones, but they are as genetically identical as you can get without cloning. And they're also a hell of a lot cheaper. So the upshot is that if you buy a strain of inbred mice, they're all genetically the same or as close enough to the same as makes no difference. And that means you can do experiments on mice that all have exactly the same genetic background. You know, if you tested some drug on a bunch of wild mice or a bunch of random mice you bought at the pet store or something like that, those are all going to be genetically different. And that's going to be a variable that affects your results. Some of your mice will be naturally susceptible to the drug, and some will be naturally resistant, and some will be smart enough to get out of their cages and escape, or something like that. If you're testing a treatment, genetic variation gets in the way. It's a variable that you can't control. If you're testing your treatment on inbred mice, all of those mice have essentially the same DNA. So your experiment doesn't get affected by genetic variation. And there's a commercial lab in the United States called the Jackson Laboratories there's their URL, jacksmice.jacks.org, that will sell you mice coming from these inbred strains. And in their lab, they breed these mice and care for them, and they'll ship you a bunch of inbred mice if you need them for a particular experiment. I mean, assuming you've got permits and things like that. And not only are all members of an inbred strain genetically identical, many strains are homozygous for genes that mimic human disease. This just happens to be a mouse from a strain called this, NOD slash she LTJ. I don't even know how you pronounce that, Nod Shilcha or whatever. Mice of that strain are virtually guaranteed to develop insulin-dependent diabetes. You do have to feed them a particular diet, but if you do that, they are practically guaranteed almost 100% to come down with insulin-dependent diabetes. If you're testing treatments for diabetes, this is a good thing because you have a bunch of mice that will all respond to treatment, that'll all get diabetes, and they'll all get it at about the same time, and they should all respond to it in almost exactly the same way. Now, my apologies to Holly, who has just joined us. I'm sorry, it took me a minute to see that there was something that I had to attend to. So I'm sorry if you missed a little bit of this. But we've been talking about inbreeding, and this is one case where heavy inbreeding, as maybe uneasy as it might make you feel, is actually used in biomedical science in the development of these inbred mouse strains that essentially don't vary and that are all homozygous for genes that can be used as models for human disease. We had a neurobiologist here 20 years ago. Long story there, but she worked with an inbred mouse strain that developed the mouse equivalent of OCD. So you can get mice that develop syndromes that look like they model human psychological disorders. You can get mice that develop equivalents of human physiological disorders. And if you go on to biomedical research, you'll encounter these, the inbred mice. Now, a guy named George Shull made this discovery in 1909. If you take two inbred lineages and you crossbreed them, the offspring are often larger and healthier than either parent. So over here on the left, the top left, you have some corn plants belonging to an inbred strain called MO17. And on the right, you have some corn plants belonging to an inbred strain called B73. And you can see that the hybrids between them are considerably larger, and the ears are also considerably larger. There again, on the left, there's MO17. On the right, there's B73. And in the middle, you have these great big ears. We see this in a lot of cases, and it's called heterosis, otherwise known as hybrid vigor. You see it in dogs. Purebred dogs might look very nice and might have certain traits that you like, but it's widely known that if you want a smart, healthy dog, your best choice is a mixed breed. Your best option is going to be a mutt, a Heinz dog. You know that expression, a Heinz dog. It's got 57 varieties. Yeah, your best bred's going to be a mixed breed pooch. Some of that's probably because a hybrid is not homozygous for most of its genes. So if there's some allele that causes a genetic defect, and it's common in a inbred strain, the hybrid is very, very unlikely to be homozygous for it. Like if I have one breed of dog that's homozygous for one disease, and I cross that with a dog from another breed that's homozygous for a second disease, the offspring's not going to be homozygous for either. There may be other things going on. I don't know that we completely understand heterosis, but it's something we've observed for over 100 years. The same's true for a lot of crops. If you've ever wondered why we don't just save the seeds from, plants that you buy at the kit in the grocery store, the answer is you could, and in some cases they would grow, but there's no telling what you'd get. You all know grape tomatoes. These have been around since 1997 is when they hit the US market. I remember a time before these existed. Grape tomatoes are in fact a hybrid. The name in the trade is Santa F1s. And the identity of the parent strains is a carefully guarded secret. So there's some company that grows two inbred strains of tomato and deliberately crossbreeds those in order to get seed that when planted will give you grape tomatoes. And because that company is the only company that knows how to make that hybrid seed, they can sell it for a sizable sum of money to tomato growers. If you want to grow grape tomatoes, you have to buy the seed from the one company that knows which varieties of tomato you have to crossbreed to get that seed. So hybridism like that can give you better crops, but it's also a form of copy protection, because think about it. Let's say I just went and bought some grape tomatoes and I saved their seeds and I planted them. Those seeds are going to be heterozygous at all, well, the tomatoes are going to be heterozygous at all loci. Those seeds are going to be an absolutely completely mixed bag, genetically speaking. They'll be homozygous at some alleles and heterozygous at others, and no two might be the same. So if I plant seeds from these grape tomatoes, there's no predicting what I'm going to end up with. Some of my tomatoes might taste great, some might taste nasty, some might die, some might be too big, some might be misshapen, no telling what you're going to get. In fact, if you go out, you could probably still do this. It's not quite too late to plant tomatoes. If you go out to a garden store in the spring and look at the tomato plants that you can plant, some of the varieties are labeled heirloom tomatoes, which means that they breed true, and others are labeled hybrid, which means you can grow the plants and they'll grow fine. But if you save seeds from those tomatoes and plant them, you're not going to get necessarily anything like what you started with. What you get might be completely unpredictable. Now, it used to be common practice for farmers to save some of each year's grain crop as seed grain for the next year's crop. So that's a poster from about 1910. There was a shortage of corn seed. There was very little of it that you could buy. So here's Uncle Sam telling the farmer to save some of his corn and save the seed and plant that seed next year. That's the way all farmers used to work. That's the way great grandma grew those awesome tomatoes that she had. She'd save some seeds from every tomato plant and then plant those seeds the next year. There are hobby farmers that'll still do this. I don't really have the space for it now, but when we lived in a house with a much bigger yard, I tried my hand at it, and it's not that difficult. You might know this. They used to run this out of the anthropology department here at UCA. They would have seed swaps every spring where local gardeners can come and trade seed from their household varieties for other varieties. So people could save seeds, trade seeds, and try growing different types of crop. Large-scale farmers don't do this anymore. Just about any corn farmer these days just buys hybrid seed from a seed company. And that started in the 1930s when farmers were encouraged to purchase corn seed that was produced from carefully selected purebred lines. Such corn was often very vigorous. This corn here is so vigorous you can see that it's turned into the golden snitch. And sorry. Oh, Harry, it looks as though you've eaten the golden snitch on the cob. 10 points from Gryffindor. Um, and anyway, that hybrid corn was not only so vigorous it could fly, but it often blended desirable traits from two different strains. Let's say you've got one strain of corn that's not disease resistant, but it's big, and another strain of corn that is disease resistant, but it's small. The hybrid has a decent chance of carrying both parental traits and being both large and being disease resistant. So hybrid corn can be healthier than either parent. And this is a major part of the reason why hybrid corn has dramatically increased our crop yields. The y-axis here is the grain yield in tons per hectare. Hectare is 3 acres and a bit. A hectare is a square, is 100 meters square, which works out to be quite a bit bigger than an acre. But anyway, in 1930, OPV right there stands for open pollinated varieties. That means varieties that were grown and pollinated, and then the farmer saves seeds and plants them the next year. And you can see yield was about 5 tons per hectare. If you go up to the year 2000, it's pushing 10 tons per hectare. We've nearly doubled the amount of corn you can grow on a given piece of land between 1930 and 2000 because of the introduction of new varieties of hybrid corn. So it means a lot more food. It's part of the reason why we have things like, corn syrup and corn sweetener in our food is because we grow so much corn that there's been pressure for food companies to come up with new things to use it for. So you produce all kinds of products out of corn these days. Everything sweetened with corn syrup because it's a lot cheaper than sugar. Well, that's also because of agricultural subsidies. But anyway, that's a little beside the point. These days we produce ethanol to go mix with gasoline out of corn. We feed corn to cattle instead of raising cows on grass. Most of your beef cattle might start on grass, but is finished mostly on corn in big feedlots. So we produce a ridiculous amount of corn, and part of the reason for that success is the adoption of hybrid varieties. The price is that farmers are now dependent on seed companies to produce the seed that they plant with, because hybrid seed won't breed true. You could plant hybrid seed, get some corn, but if you plant seeds from that batch of corn, what you get is completely unpredictable. Okay, so that's what I wanted to say about hybrids, and what I wanted to say about some uses of hybrids in agriculture. Okay, real quick. We've already talked about genetic drift, and I gave you that demonstration on Monday of what it looks like in simulation. Genetic drift results from random effects in a population. It's what happens when one member of the population, for reasons that have nothing to do with selection, just happens to have extra offspring or just happens to fail to reproduce. In a large population, each random event only has a tiny effect, and many of them will cancel each other out. So if the population is large, random events do very little. In a small population, random events have a much larger effect. Um, if one person dies in an accident before they can have kids in Little Rock, that has almost no effect on allele frequencies. If one person dies before they can have kids in Possum Grape, Arkansas, or Booger Hollow, Arkansas, that has a much larger effect because the population is small. And if you remember, we did some of these with that PopGen simulation that I showed you. When the simulated population size is 1,000, you can see that over the course of 50 generations, allele frequencies stay pretty close from where Hardy-Weinberg would predict them. These colored lines are clustered together. When the population's 20, they zig and zag all over the place. And we saw that again on Monday. So qualitatively, you can see what genetic drift tends to do. I did include some equations. Do not memorize these. You don't have to know them. I want you to understand them qualitatively, but I'm not going to make you solve them. They won't be in problems on the test. So we often want to know how much effect genetic drift is going to have. It turns out it depends on what we call the effective population size. A real population has a certain size. It's got a certain number of critters living in it, but many of those members may not be breeding. They might be too young. They might be too old. They might be sick. They might, any number of things could have happened. So the effective population size is an ideal population size, and it's the size of a population that would have as much genetic drift as the real population that you're studying. So you can measure genetic drift in a real population and calculate the effective population size. It's written N sub E. And if you have a population that includes breeding males and females, it's equal to this. It's 4 times the number of breeding males times the number of breeding females divided by the number of breeding males plus the number of breeding females. There actually should be some parentheses right there. I forgot to include them. Don't memorize that equation, but let me see if I can make this a little bit more clear. If everybody in the population can mate, the effective population size is close to the real population size, but we don't always see that. These are a couple of male northern elephant seals. Male elephant seals are about twice the size of females, and each male defends or tries to defend a harem of females. So a really big bull male elephant seal will have 10, a dozen, 20 females that he alone mates with, and he has all of their babies. Now the problem is that there are equal numbers of male and female elephant seals. So if one male has 20 females, 19 males have to sit in their basement and play Among Us, or whatever it is that male elephant seals do when they can't get a date. All right, whenever you have one male that's guarding multiple females, that means multiple males are left out in the cold. Incidentally, this sets up some absolutely savage fights because a male that, the only way for a male to breed is for him to take over another male's harem, upon which he will immediately get to reproduce with all of the females in the harem. And this is why males fight absolutely viciously, slamming into each other with these bodies that can weigh over a ton and stabbing each other with their canine teeth. You can see the scars on the necks of both of these. Males often fight to the death in northern elephant seals. And so in a northern elephant seal population, where there's only a few males that mate with almost all of the females, the effective population size may be remarkably small. Let's go back here. If we have 1,000 organisms that are all interbreeding freely, we would expect to have a situation like this. Allele frequencies aren't going to change very much over time. But we could have a population of 1,000 elephant seals, but the effective population size would be a lot smaller than that because only a very small number of males are having almost all of the sex. So, momentary flashback to high school there. So we might have a population of 1,000 elephant seals and yet see this kind of zigzagging if we track allele frequencies over time. There might be 1,000 elephant seals, but they have as much drift as you would expect if there were only 60 elephant seals. The reason that matters is, look at this. We have these zigzag lines that show us allele frequency. 2 of them, here and there, hit the 0. What that means is that, the frequency of a certain allele went down and it zigzagged, but if it hits 0, it's never going to come back up. Once the frequency of an allele hits 0, that allele is lost. It doesn't exist in the population anymore. So that means that even if you have a large population of elephant seals, allele frequencies are going to zigzag as if the population was a lot smaller. And the more they zigzag, the greater the probability that alleles are going to get lost. And your population of elephant seals is going to end up being less genetically diverse over time. That's something that ought to concern wildlife managers, because genetic diversity is one of a population's defenses against going extinct. If you have a population where everybody's identical, then all you need is one bacterium that can kill one of them, and it can wipe out all of them, because if they're all identical, none of them are going to have defenses. If, on the other hand, you have a genetically diverse population and a bacterium hits that, some new infection or virus or whatever, and that new infection hits, if the population is very variable, the greater the odds that some of them are going to happen to be resistant to that bacterium or virus or what have you. So the more genetic diversity you have, which just means the greater the number of alleles in a population, the more resistant it is to certain things like epidemics that could drive it to extinction. So you want lots of alleles in a population that you're trying to conserve. And if you have a large population of elephant seals, but you have genetic drift like it's a small population, you're going to start losing alleles over time. That population will become less and less diverse. It's also critical in livestock management. Back in the old days, if you had cows and you wanted to have baby cows, you brought in a bull, and the bull did what bulls do and mated with all of your cows, and then all of your cows would have calves and they'd grow up, and the great circle of life. Yeah, that's the old way of breeding animals. By the way, it's still legally required in thoroughbred racehorses. A thoroughbred must have been produced by mating in the expected way. The stallion must cover the mare, as they say. But with an awful lot of livestock now, you can do artificial insemination. If you've got a champion bull, you can just collect that bull semen and freeze it and ship it anywhere it's wanted. And if you're a farmer, you can buy frozen semen from a champion bull and thaw that semen out and load it in a device called an insemination gun, which sounds really scary, and go around and impregnate your cows. Now, that means that if you're a farmer, you have access to much higher quality bulls than you'd ever have if you just had to, you know, buy one or borrow from your neighbor or something like that. You have much higher quality bulls. The problem is that we now have in cows what we see in elephant seals, where a very small number of bulls are the fathers of tens of thousands of offspring, because each high quality bull might produce, I don't even know how much semen he's going to produce, but he will produce enough to impregnate tens of thousands of cows and produce tens of thousands of calves. And the top dairy breed right now in the US is the Holstein. There's a Holstein, reminding you to eat more chicken. And there might be hundreds of thousands of Holsteins in the country, but the effective population size has been calculated to be about 80. And the reason is, even though there's hundreds of thousands of Holstein cows, almost all of them have been produced by a very small number of elite bulls. A small number of the very best bulls are doing all of the breeding, and all of the male cows that are not in this elite squad don't get to breed. They probably end up as dog food or something like that. And that effective population size has been calculated at about 80. That means that even though we have lots of Holstein cows, the Holstein cow population is subject to a lot of genetic drift. It will behave over time, as far as drift goes, as if it was a small population. Allele frequencies are going to zig and zag all over the place. The reason we are worried about that is, again, the more genetic drift is going on, the greater the odds that an allele is going to be lost. The greater the odds that it will end up as zero. You could compare allele frequencies over time to a drunk staggering down a street. So this takes me back to my undergrad days in New Orleans, where we had drunks staggering down the street on any given night, including my roommate freshman year, but never mind that. So if a drunk is staggering down the street, he might stagger to the left, and then he might stagger over to the right and zigzag to the left and zigzag to the right. If he hits either one of those gutters, he's going to fall in and stop moving. Same thing with allele frequencies. Alleles might become common. They might zigzag closer and closer to 1, and then become rare and zigzag closer to 0. They're going to zig and zag every which way. But if an allele frequency hits either 0 or 1, the allele frequency is going to stop changing, right? If the allele frequency hits 0, it means the allele is lost. If it hits 1, it means the allele is fixed, meaning it's the only allele left in the population. And so if we go back here, with a large population, in all 5 of these populations, both A1 and A2 stayed in. In this small population, we did 5 different populations, and in 2 of them, the A1 allele was lost. And in a third, the A1 allele was very close to 0. So the more genetic drift is going on, the greater the probability that you're going to lose an allele. This is how we calculate it. The variance in allele frequencies is a measure of how spread they are, and it's going to be equal to PQ divided by 2 times the effective population size. Don't memorize that. The length of time it takes for an allele to be either fixed or lost is this. And all I want you to see is that it's directly proportional. Never mind, it's inversely proportional to, no, it is directly proportional to effective population size. That's NE right there. If NE is very large, then it will take a very, very long time for an allele to be lost or for an allele to be fixed. On the other hand, if n sub e is small, then it takes much less time for an allele to be either lost or fixed. And again, this is the kind of thing that wildlife managers have to be concerned about, because when you're preserving an endangered species or a rare population, you want to keep as much genetic diversity in it as you possibly can, um, for the simple reason that the more diverse that population is, the likelier it will be able to withstand environmental changes and epidemic diseases and things like that. But if there is a lot of genetic drift going on, um, you may be in trouble because that means that alleles are much more likely to get lost over time, purely by drift. That's actually a good place to stop. So I'm going to go ahead and stop the recording here.